Okay, so where was I? Okay, so I talked about writing down a likelihood for an individual's genotype given where they're from. And so using this likelihood, we can go to Bayes' rule, which is a basic probability rule, to estimate, to compute a posterior probability that an individual comes from each of the K populations. And so here I'm just writing out a formula that comes from this standard rule of probability in our own terms. So basically it's saying that the probability that an individual I comes from subpopulation K is going to be equal to the probability of seeing their genotype, given that they come from population K, times the prior probability that they come from population K. And then that's going to be divided by the denominator, which is the sum of this thing across all populations. So for each population, we're going to compute the likelihood of seeing an individual's genotype there times a prior probability that they come from there, and then that's normalized by the same thing looking across all the possible places they could have come from. And so let me just say something about this prior probability. So the prior probability in Bayesian statistics represents our prior information about something we're trying to estimate. So in this context, if we had an idea that somebody might come from a particular location, then we might set this prior probability to be high in that particular location. So most in the default version of structure, we assume we don't have any initial information, so we just set this to be equal to 1 over k, so it's just symmetric across all the populations. So this actually just completely cancels out. In a little bit, I'm going to talk about these migrant models, where we have an idea that that an individual is probably from a particular place, and then we want to ask whether that might be wrong. And so in that case, that's where we'll start modifying this.
Yeah. So it strikes me that that's a nice null prior.
Yeah.
You've got equal numbers of individuals in all your populations. But if some of your populations are bigger, but then you don't know that, and some of them are really small, then 1 over k is actually the prior is actually going to be influential on the posterior.
Yeah, that's a good point. Yeah, so we could try to estimate. So you can imagine having a hyperparameter in the model that tries to estimate that some of the populations have actually got a lot more individuals coming from there than others, and then that would help you to do a better job of assigning individuals where you're not really sure. And in the Admixture Model, we have a version that actually does try to do something like that. When the model's working well, the genotype likelihood is really informative, and so the exact way you set the prior doesn't really matter too much, which is why we don't worry about it. But if you're on the margin, then these kinds of things you're talking about really do matter. So we have done various things with playing with these priors. There are certain settings where doing a better job with these priors really makes a difference, but sort of a minority of the cases. Anything else on that? Okay, so just to summarize that, this is basically saying that if we G is the observed genotype, and then P is the allele frequencies in the population. So if we know what the allele frequencies are in each population, then it's a straightforward computation to figure out the probability that an individual comes from each place. Okay, but to compute that, we need to know what this allele frequency matrix P is. And then similarly, a natural estimate of the allele frequencies in in a particular population is easy if we know where individuals are from. So you can imagine in the no admixture model, if we want to estimate the allele frequencies in subpopulation K, then all we're going to do is to look at who are all the individuals from subpopulation K and just count up how many copies of each allele they have, and and that's your estimate of allele frequencies. And again, in the Bayesian model, we do something which is slightly more complicated, where we have a single Dirichlet prior. You don't really need to worry about that complication too much, unless you're interested. Okay, so so those two steps really are very simple. They kind of come out of this like the assignment literature that I was describing from the 90s. But when we don't know either the allele frequencies or the population assignments in advance, then it becomes a little bit harder, and so we developed this Markov chain Monte Carlo algorithm to get joint estimates of both allele frequencies and assignments at the same time. So here's the Markov chain Monte Carlo algorithm. So we start out with a random assignment of individuals to populations. So those are the z's. So we're assigning the z's at random. And then, in step 1, we take those random assignments and estimate the gene frequencies in each population. Step 2, taking those allele frequencies, we reassign individuals to populations, and then we just repeat this a lot of times. And so Markov chain Monte Carlo is sort of a well-known statistical procedure, and the way that it works is that you start out by from some arbitrary or random starting point, and you run it, and it sets up this stochastic process that starts out in a ridiculous part of the parameter space, but there's this theory that shows that eventually you get to a really sensible part of the parameter space, and then this as a probabilistic model, so it spends time exploring the parameter space. And so the standard way that people run MCMC algorithms is that you start out with what's called a burn-in period. So the burn-in period are these initial steps that allow it to get away from this silly starting point into somewhere sensible. And then once you've decided that you've got to a sensible place, then you just track the behavior of the Markov chain as it explores the posterior distribution. So it's basically, you can think of that basically as being like exploring the space of possible parameter values that are consistent with your data. So one thing of this, so the nice thing about this algorithm is it's allowing us to estimate the joint distribution of all of these unknown factors at the same time. Secondly, MCMC is really powerful because it allows you to estimate how much uncertainty you have in all of your parameter estimates, because the Markov chain kind of explores around the space of plausible models. Questions? Oh, and this is all for a fixed value K. Okay, so up to now I've talked about what we call the no admixture model, in which each individual is derived completely from a single population. But early on we were really interested in cases where individuals have got mixed ancestry. And so even though it's more complicated, the first thing we actually worked on was what we refer to as the admixture model. And in the admixture model, we assume that each individual has got basically a vector of ancestries which represent what fraction of their ancestry comes from each of the K populations. So for example, you might have ancestry 1/4. So let's suppose you've got one grandparent from population 1 and 3 grandparents from population 2, and then no ancestors from any of the others. Then your ancestry vector that we would like to estimate would be 1/4, 3/4, and then zeros. And so then the way that we interpret those ancestry proportions then is that if you've got ancestry proportion Q sub K, then that means that a fraction Q sub K of your genome should come from that population. And so your allele frequencies in that segment of your allele frequencies in those alleles should follow the allele frequency distribution in population K. So we find in general that the no admixture model is usually a little bit restrictive. We really prefer the admixture model. It seems to, it's a lot more flexible. We think that it fits real data better. Real data, of course, are much more complicated even than the admixture model, but we think this is quite a bit better than the vanilla no admixture. So one of the first data sets that we applied this to is this thrush from Kenya, and this is an early study that was published in 2000. They collected data for 7 microsatellite loci from 155 individuals. There were 3 sampling locations. I think these are forest refugia, and they're separated by agricultural land. So perhaps they think that it's unlikely that birds will be moving freely between the 3 locations. So one thing that we did early on, which was not using structure, was to build a neighbor joining tree of the microsatellite data. So this is analogous to the tree that I showed you for the human data early on. And so basically to make this tree, what we're doing is building a pairwise matrix of microsatellite distances between all of the individuals, and then sticking it into a tree building program. So you can think of this really as doing clustering of similar individuals rather than being analogous to a true phylogenetic tree. And so they I think some of the colors have gone wrong here. Anyway, okay. So I think the colors haven't been rendered correctly. There are two different reds. I'm not sure why that is. So all of those individuals there, almost all those individuals there, are one population. That's a different sampling location than the fruits you can see. So this neighbor joining tree already shows you that there's fairly there's clear evidence for separation of 3 groups. But if you just had this without the labels, it would be very difficult to actually tell that there are three groups, and it would be very difficult to you wouldn't be able to see where the 3 groups are. So the first question is whether we can use structure to detect the fact that there are 3 distinct groups here. And so what I'm going to show you is it's kind of a time-lapse movie of the Markov chain Monte Carlo process of analyzing these data. And so early on, I don't use this triangle representation so much for the ancestry proportions now, but we used to use it quite a lot for handy when there are 3 populations. So basically, in this triangle, so for each individual, we're trying to estimate an ancestry vector, which sums up to 1. So it's the fraction of each individual's ancestry from each of the 3 populations. And so, so every point in the triangle represents a possible. Every point in the triangle represents a possible ancestry vector that adds up to 1. So if you're in one corner, that means that your ancestry proportions are 1, 0, 0. And if you're in the middle, that means that they're a third, a third, a third. And so this is showing you just that the initial configuration, we've assigned individuals completely at random in this space. Okay, so now, after 25 iterations, it's starting to spread to sort individuals according to their genotypes into the towards different parts of the triangle. And the initial directions in which it sorts these are really random. So I think that right in the beginning there must be some slight asymmetry, random asymmetry that slightly more of the individuals from this population are located towards population 1, and then it starts estimating the allele frequencies in population 1 to be a little bit more like those, and then it's sort of that initial asymmetry starts off pushing it as a runaway process in one direction or another. So this is after 25 iterations. Here it is after 200 iterations. You can see the individuals are slowly moving further towards the corners. 400 iterations, 100,000, and so this is the average over 100,000 iterations after throwing away some of that burn-in period at the beginning. And so now you can see that with using just 7 microsatellites, most of the birds are assigned quite strongly into one corner or another. So there's the blue population here, the red and the green, and then there's a small number of individuals who look either like they're migrants or they're misclassified. So perhaps that blue individual here, that's a blue individual there. So this shows then that they, with 7 microsatellites, we can assign individuals fairly well. So there are a number of questions that this raises. So one is K equals 3 the right model? A second question is an individual like that a migrant? So perhaps this individual that was sampled in the Ngango population is actually from Chawea. So can we get strong evidence from the genetic data? Can we assess the evidence from the genetic data for that model? So the first question is estimating K, and so the way that I should say that estimating the number of clusters in these mixture models is well known to be it's just a really hard problem, and it's hard it's difficult from a technical point of view, and from a technical point of view we've got some approximations in the math that allow us to compute something that's a little bit like a penalized likelihood, but also there's kind of more of a fundamental challenge in estimating the number of populations, which is that usually when we're fitting this to real biological data, the true structure is much more complicated than our toy model. And so, if you knew everything about a population, there might not be a natural value of K that you would say, this is the correct value. As I said, I work in humans a lot, and if you think about, let's say, human population genetic variation across Eurasia, there's a lot of isolation by distance going on. There is some amount of, there's some locations where you've got barriers to gene flow. But overall, if you apply structure in a setting like that, it's really unclear what kind of value of K you would like it to return. And so I think that there's been way too much emphasis in the literature on trying to come up with what's the best supported value of K. And so I think it's important to have some criteria for thinking about what's a sensible value. But I really don't like the emphasis on trying to come up with a true value, because I think most of the time it's not really a very well-defined quantity. So in this case, with the thrush data, we applied this method that we developed for estimating K. Oh, I should just say, so if you think about a maximum likelihood model, as you add parameters in a maximum likelihood model, the fit always gets better. But in a Bayesian model, where you have these, where we're sampling from the posterior distribution of the parameters given the data, as you add populations, what happens is that the number of individuals assigned to each population starts to get smaller, and so the allele frequency estimates become more uncertain, and so that actually winds up dropping the posterior probabilities. And so that's the kind of thing that we leverage to try to estimate a posterior probability of K. So you can think of it as sort of like penalizing models that are unnecessarily complicated. So in this case, the data are very clear that the 1 and 2. So this table is showing the different possible values of K, what's the posterior probability of these different values, and the data are very clear that 1 and 2 aren't sensible models. It prefers 3, although you could add additional populations, and maybe there's a tiny amount of support for having 4 populations. I think that this thrush data set is probably unusually easy from the point of view that its sampling is from these very localized and very discrete refugia. So as I said, I think in most settings it's less clear what the answer should be. All right, I think I already said most of this. Yeah, so, I mean, another point is that I think that structure is often most useful in terms as a kind of a visualization tool for understanding what patterns of variation look like. And you can kind of imagine if you have, if the reality is complicated and you have a lot of data, then the true value of k that you estimate might become arbitrarily large as you add more and more data in terms of both numbers of individuals and numbers of loci. And as k becomes very, very large, structure actually becomes less useful as a visualization tool. So I think that it's, you know, I'll show you some examples in a minute. I think it's often most useful to use structure as a way of kind of look at how the clustering, look at the clustering of individuals, the different values, a whole series of different values of k, and it kind of gives you some gestalt notion of what does population structure look like in this population, rather than the approach of picking a particular value of k and reifying that as as the truth.
I think it's very interesting what you were presenting, because also brought back to the discussion yesterday the practical value of the inference, k inference. I think the practical value of it, well, you didn't envision this when you created the program, but I think the most practical value today is try to compare how many populations you have, how many evolution and significant units you have to manage this. That's critical. So they really want to know what is the most likely value of k.
Right.
So
yeah, okay, that's a great point. So I think that what it can do well is tell you that like these individuals over here are really different from these individuals over here. But I think that they, like in most cases, that like there is no true value of k that would make sense. So if you collected your samples and calculated that k equals 4, you're really confident about that, I would guess that if you went out and collected 10 times as many samples and got genome sequences for all of those and reran it, you should probably get 15 populations. So they, I think that population structure is it tends to be hierarchical, and like from very local scales to larger scales, and I think that I guess that there's some sort of experience with how structure behaves when you've got some moderate number of microsatellites and realistic current sample sizes, but it's not really estimating something real most of the time, because if you increase the data, you get more structure. I think that management is sort of a bigger question, like how do you set the how do you decide what's a reasonable amount of differentiation you want to require set conservation. I mean, you guys are in this field. I'm not really, so I'll defer to you. Yeah.
I so agree with you that there's value. There's really, you run structural one, you see different cases, and there's information you can get from them that's useful.
Yeah.
Right? You try to get them past reviewers. They will say, use Ivano et al. You can determine 1k and all other k's are wrong. I mean, I've gotten this multiple times, and it's really difficult to, because there's no other reference I can use. If I could just say, Pritchard et al., says that you should look at all, at the other case. That would be really helpful because it's in the literature people, just focus on this one K value, and we can write back pages of responses and say it's unreasonable because that's always work. But it's very hard to sketch.
Yeah. Well, maybe we should write something.
Just cite ConGen.
No, I'm serious. I don't. I guess I wouldn't feel like doing it entirely myself, but if you or others would like to work with me on that, I'd be up for that. Yeah.
So Jonathan, you said that you might have a certain data set and see that K equals 4 seems to have a very high posterior, but then go and collect a lot more information in the genomic sequence that you might find that K is 15. But the 4 that you found originally would still be superimposed on that structure of 15, right? So,
yes, exactly. Yeah. So I think it's like the structure tends to be hierarchical, so that as you add data, you find that what looked like a homogeneous group now you can separate it out. And I'm going to show a great example of that in a little bit, where there's a recent paper. I won't give away the punchline, but there's a recent paper from another group that shows you just incredibly fine structure. Okay, this is great. Any more thoughts on that? Yeah, go on.
I was just going to say that was a really helpful comment, that it's up to you guys to decide what's important or meaningful, and it links to Robin's lecture, which is next, identifying management units and differentiating statistical significance from biological significance. So I think that's helpful.
Yeah, thank you. Yeah, I actually think that's really. I'm really an outsider to this field, but it seems like that's a fundamental question. It also ties into the discussion last night. I've no doubt that as this field moves towards collecting genomic data, you'll find more and more and more structure. And the structure is real, but it's really fine-grained. And at what level is it realistic to do conservation? I guess you guys who are on the close to the management side have to figure that out. So let me just run quickly through some bells and whistles that we developed for various kinds of settings. So this is getting to your question about the priors. And the first kind of, just to remind you, one of the kinds of priors that we had in the model that I showed you before is that there are K populations, and we're going to assume initially that every individual is equally likely to come from each of the K populations. But let's suppose that we want to do something like detecting migrants. So in the thrush example, you've got one of the birds that was collected in the blue location, and its genotype actually looks more like the genotypes of birds from the red location. So should we conclude that's really strong evidence that that individual is a migrant? And so to tackle that kind of question, we've developed these models where we set a prior that's informative in the sense that we say that individuals, we say that the clusters, the subpopulations will be guided by where the individuals were sampled, and those will usually be correct. So, okay, that's a lot of text there. So basically, the model is that we're going to so in this migration setting, we're going to assume that we have a good idea in advance of what the populations are. We're going to assume that those are usually correct. And so we might say that there's perhaps a 90% or a 95% prior probability, which means sort of like your expectation, that an individual will come from the location where it was sampled. And then the remainder of the probability represents the possibility that that individual is either a migrant from somewhere else or they have some recent migrant ancestry. And there are a lot of different ways you could model this, but the particular choice that we made was we assumed about like all the different scenarios through which somebody might not be purely from the location of their And so now we can go back to the thrush data, and so there were 4 individuals on this picture who look like maybe they don't really belong with their own population. So, for example, there's this blue individual here. Actually, their genotype looks much more like the red. So that's individual number 1. So they were sampled here in Ngango. We're going to propose that the possible source population is Choea. And now this parameter nu is representing the I guess a prior probability that they came from somewhere else. So so if nu is 5% or 10%, that means that we think that there's a 5 or 10% rate of people of birds coming from birds having migrant ancestry. And so this parameter nu is kind of arbitrary. We often wouldn't really know what that is in advance. But the way that we thought about this is that we could try a bunch of values of this parameter. And if the data are really, really clear that the individuals are migrant, then you should have a high posterior probability that the individual is a migrant, no matter what kind of value you set nu to, as long as it's sensible. Basically, the take-home message from this was that in this particular data set with only 7 markers, we can't really be confident that anybody is a migrant. So, so for example, for this individual number 2, we're estimating that there's about a 50% probability that they have no immigrant ancestry. So 67% with this prior and 47% with this prior. And then if they do have immigrant ancestry, then it's unlikely that they're a first-generation immigrant. It's more likely that they have an immigrant parent or perhaps a grandparent. But in this particular setting with relatively few markers, this is saying that we really don't have enough data to be able to detect migrants with any confidence, even though these individuals kind of look like they might be outliers. With modern data where you have more microsatellites or even better genome-wide SNP data, then you can be extremely confident about migrants or individuals with migrant ancestry. So the migration model is kind of fundamentally different from the basic structure models, because in the migrant model we're sort of telling them what we think the answer is, and then looking for whether there are individuals where there's really strong evidence that our prior prediction was wrong. And so it generally takes a lot of data it takes really strong data to be able to overcome the prior expectation. So another setting that we have looked at is a case where the population structure is actually real, but it's very weak, and so structure has a hard time finding it. And so the reason that we started working on this model is as follows. So a lot of people told me, I sampled my organism in these 3 places, and I compute FST, and a significant FST between the groups, and then I run structure, and structure doesn't find anything. So structure really sucks. And so the way that we thought about what's going on here, and the way we thought about this is that like structure's trying to do something that's, like it's trying to solve a problem that's kind of harder than the FST analysis. So in in that FST analysis, you start out with these predefined groups, and you ask whether they're significantly different from each other. But the standard structure model is starting out with no information at all. It's got no idea who comes from where, and then it has to be able to group individuals and be confident that the group, in some sense, that the groupings are real. So there are a huge number of different ways that you can assign a sample of individuals to K groups, and structure in effect puts a uniform prior probability on all of those enormous number of combinations of ways of assigning individuals, and so that kind of acts like a penalty against believing any particular combination of assignments too strongly. And so the idea that we had was that we could investigate models where we give structure an idea in advance of where individuals are sampled, and it's basically saying, if you want to go ahead and make these sampling locations be similar to the population labels, if that's supported, then go ahead and do that. But if the population structure doesn't match the population labels, then ignore it. And so I had a student in my lab, Melissa Hubis, a few years ago, who basically developed this prior model that's taking in the population labels and doing exactly this. So if the population labels really do look like the structure, then it uses them, and if the structure doesn't follow the labels, then it ignores it. And it really makes a big difference. So there's in this set of this part of the parameter space where the signal is structured, but the program structure isn't really able to get it, then this model can often kind of push you over the top. Of course, it's a little bit more aggressive with the data, so there might be some. One thing we worried about a lot is the possibility that it might kind of overinterpret the structure labels, and Melissa did a lot of simulations of this, and it seems like it works pretty well. It's pretty robust. So yeah.
Given your result for a certain p-value without using the forward priors, would you recommend reporting that then instead of using the priors?
Yeah, I think it's easier to interpret if you don't use them. Like if you get really clear structure without using the labels, and particularly if what you infer kind of matches the geography, then it's sort of a slam dunk that it's finding something real there.
If it's just slightly improved with the priors, but then maybe
you could show it both ways, but with this model, I always have Melissa did a lot of simulations, so I feel pretty good about it, but it always sort of nags in the back of me, could we be overusing the prior a little bit? So if you don't have to use it, I probably wouldn't, but we think it generally works well. Yeah.
But isn't that statement you just made fundamentally anti-Bayesian?
Which one?
That if you have information about how something might be, you ought to use that information in the first place.
That's a great question. So there's.
But the data will overturn the prior if you've got enough of it, right?
Yes, that's right.
You can imagine a circumstance where those priors are bounded.
So I think that there's kind of a debate between the pure Bayesians and the pragmatic Bayesians. So I think of structure as being kind of pragmatic Bayes. Most of it is not using very strong priors. I think the basic model is saying, we start out with no information at all, and then if you get some clustering out and you see that it matches the if you see that it matches the geography, then because you held out this key information, when you see that those match, then that really makes you believe it. So the pure Bayesians, they really, really totally believe their models. But the pragmatic Bayesian might say, well, I believe my models most of the time, but I'm not sure if I'm willing to bet the house on it all the time. And so I hold this bit of information out, and if they agree, now I really believe it. So yeah, I think this point is debatable, but I sort of come at it from kind of this pragmatic position. Yeah.
What if you just throw more data at it? I guess it costs money to generate sequence data.
Yeah, well, that's your guys' job.
So Jonathan?
Yeah.
Going back to where you started, just because you sample 3 populations and you get significant FSTs, doesn't mean there really are 3 separate populations. There's all other more complicated things that could be going on. Why you could never find a decent structure.
Yeah.
5 populations, they just happen to be non-randomly distributed where you sample.
Yeah.
Or maybe there could be 3, but there's so many individuals moving around.
Yeah, I completely agree with that. So so I think that this is kind of more or less useful depending on the details. So one kind of scenario could be, let's suppose you've got 10 individuals in each of 10 different locations. Then what this model will do is it'll tend to hold each of those locations. There'll be some tendency to hold each of those locations together, although it won't be required. So if the 10 locations actually represent 3 underlying populations, then it'll still have more power to detect the 3 because it's kind of moving those individuals together. You can think of them as being sort of super individuals, and and then at the same time, if some of those sampling locations actually don't really follow the structure there, they're just kind of random, then it should in principle be able to find that. So I think that there are. If you have 3 sampling locations and it just estimates that k equals 3 in some way that it's not really all that useful. Because it's part of what you're getting to. But I think sometimes it can just kind of bump you over the top. And it also gets to the it helps people feel better if they believe that there's something there and structure's giving them nothing. Yeah.
So is this model more sensitive or has more trouble with isolation?
Oh, that's a good question. I don't know. In general, structure doesn't. General structure's trying to shoehorn isolation by distance into the discrete model. So I don't know if this would be better or worse than that. Just think about that. Yeah, Robin.
And what's in the y-axis on your graph?
Oh, I'm sorry. So this is a measure of. So these are simulations, and this is a similarity between the estimated population assignments and the truth, and so I think if you don't have any information, then it's probably just 1 over k, and then there must be some penalty to make it go down to 0. So what this is supposed to show here is that the black is the accuracy when you have so this is for some fixed sample size and number of loci. So the black is the accuracy when you have when you're just using the uninformative prior model, and then so basically there's this transition zone where for these particular parameters between FST of about 2% and 4%, where it goes from over replicate simulations, like at 2% it basically never gets it right, and at 4 or 5% it generally does get it right. And then if you give it the prior information, then at much lower levels of FST, it's generally getting it right. Yeah, sorry, I got excited. I forgot to explain the graph. There's actually some theory that Nick Patterson colleagues developed, which looks at basically there's sort of this threshold behavior for these clustering models where you switch from being unable to model the structure well to being able to get everything right. And you sort of see that here. So the threshold is is a function of FST, number of loci, number of individuals, and so as you increase some multiplicative combination of those things, like you get to this point where you can get it right, and then when you're above that threshold, then like over repeated sampling of the data, you sort of consistently get it right, which is sort of what you see here. So let me just run through these quickly. So one of the early applications of structure was in a 2002 paper where we were looking at data from humans from 52 different populations, about 1,000 individuals, and there were data collected for 377 microsatellite markers, and let me just step through these. So these are the 52 different populations, and they're ordered geographically: Africans, Europeans, and so on, Native Americans on this end. And then each individual is represented as a thin vertical colored line, and the colors represent the fraction of ancestry that's assigned to each of the K groups. And so it's sort of a commonplace now, but at that time I think it was exciting for us that we were able to really cluster individuals according to continent quite well. So this is a Sub-Saharan African cluster. This is a cluster that's comprising most of Western Eurasia, so Europeans, Middle Easterners, and Central Asians. This is an East Asian cluster here. These are Oceanian populations, so Melanesians and Papuans, and then there's a Native American group here. So there are some interesting things that this plot illustrates. So one is that at k equals 6, there's an isolated population in Pakistan and Afghanistan called the Kalash, and they pop out as a cluster. And this isn't a totally consistent result, so different data sets on the same samples don't always show the Kalash popping out this early. But one thing this really illustrates is that we don't think that the Kalash super diverged from other populations, but they are relatively inbred. And so because so we think that the homogeneity within the Kalash leads them to be sort of popped out at this stage, and so I think that showing these, like sort of stepping through these plots for different values of k, I think it is really useful. But the order in which structure breaks out the clusters depends on various things that, I mean, not all of them which are what you want. So it depends on how big the clusters are and how diverged the clusters are, but also like if you have a cluster that's really like it's a little bit inbred, then it might prefer to sort of pop those out earlier. So now just got timing on it. Let's see. You also see admixture in this clearly. So for example, this population here is the Mozambique, who are they're the sorry, I'm blanking on their name. They're from Algeria. They're mostly they're mostly sort of Western Eurasian, but then they have this they have the Sub-Saharan African component. In the Maya, you see that they have a European component, which is presumably from post-colonial admixture. Since this is quite a large and complicated data set, instead of continuing to increase k past 6, we found that it was most useful to do analyses within the different continents. And so here in Eurasia, you can see that there's a European group, there's a Middle Eastern group, a Central Asian group, and again the Kalash pop out. So in a data set like this, where there really is a huge amount of population structure that's represented, we've generally found that it's helpful to sort of do the analysis hierarchically rather than trying to go up to a huge value of k with everybody in the data set. Should we take a 2-minute break, and then we'll keep going? All right. This hypothesis that canine CTVT, canine transmissible venereal tumor, come from a single source, and what breed or geographic location is the origin. They were also interested in trying to date the origin, and that turned out to be kind of difficult. I won't talk about that now. So they had an analysis with a whole bunch of with about 70 microsatellites from the tumor samples, as well as from a number of whole bunch of dog breeds as well as wolves. And so initial structure analysis that I did showed that the tumor samples were most similar to a cluster of Asian dogs as well as wolves. And in fact, it seems that the CTVT here clusters in with wolves in this analysis. And you can see a neighbor joining tree as well that's showing, seems to be showing the same thing in a different way. So our best guess is that this tumor may have come in from the wolf population and into the dogs. I wanted to talk a little, just for a moment, about genome-wide data. So now that many of you are moving towards genome-wide SNP data sets, the original structure algorithm is a bit slow for these. Although one point I would say is that some of you are telling me that you run structure for extremely long runs, and I think generally that's overkill. But there are a couple of algorithms that have been developed that are much faster than the original structure. So Admixture is from a group at UCLA, and that's a maximum likelihood method. So instead of trying to explore the posterior space, what Admixture is doing is just trying to maximize that likelihood function, and it does that really fast. Fast structure is from our group, and it's a variational Bayes method, which means that it maintains some of the properties of the Bayesian approach, but it's quite a lot faster than structure. So in terms of speed, it's between structure and admixture. I wanted to tell you just a little bit about a program that was written maybe 3 or 4 years ago by Dan Lawson and some other people in Britain, called Fine Structure. And this is a really powerful method that they've designed for looking at data where you've got dense SNP data, and you also need to know where the markers are. And so their idea was that they start out by doing what they refer to as chromosome painting. And so basically the idea is that if you imagine you've got a whole sample of individuals and dense SNP typing data. So then for each individual, you can ask, as you go along the chromosome, who's the most similar other individual, other haplotype in your sample? And so basically for what you're doing is you're trying to paint the haplotypes for each individual according to who are the most similar neighbors at each location. So if you can imagine, like as you move along a chromosome, that then this brown individual may be the most similar chromosome for the first stretch, and then there's an ancestral recombination event that occurred, and then there's a stretch where you're not quite sure who the most similar other haplotype in the sample is, and then there's another recombination event, and then it's copying from this purple individual. And I'm sort of racing through this, so this is probably not very clear, but basically the idea is that for each individual, you could they get what they call this copying matrix, which tells them who's the, like, as you go across the genome, who's most similar at each location. And so that kind of brings in haplotype information, which has generally been ignored in the vanilla version of structure. And so they make these pairwise matrices of similarity, and what they claim is that they can really detect very, very fine scale differences in similarity between groups, and they can get a lot more resolution than the original structure. And so there's an application that was published earlier this year applying this method in the British Isles, and I think the resolution here is really fantastic. So this is a sample of about 2,000 individuals from around the British Isles, and they chose the individuals quite carefully. So the requirement is that all 4 grandparents had to have come from the same place, and so each symbol on here represents a sampling location of an individual, and then the colors represent assignments to clusters. And so I think this is about 15 or maybe 15 to 20 different clusters here. And so they're not using the geographic information, but you can see that even for humans within the British Isles, they're able to find incredible structure. So I don't know how many of you are familiar with British geography, but so for example, they're able to distinguish people from Cornwall from people from Devon, which are neighbouring counties in Britain. There's a large cluster which is sort of southeastern England, which is where I grew up. Not that my grandparents came from there. Anyway, there's a distinct cluster in Wales, and there's a strong boundary between England and Scotland, which makes the Scots happy.
And what about Ireland?
Well, so they didn't sample Ireland proper in this, but so there's similarity between Northern Ireland and the west coast of Scotland, which makes historical sense because there's been a lot of gene flow, a lot of migration back and forth. So I think that this plot kind of gets back to some of the points we were talking about a little earlier, that as you add more and more data, you'll find more and more structure. 10 years ago we thought we were doing pretty well when we could distinguish continents, and now we can distinguish English from Scots and Devon from Cornwall.
What about the Orkney Islands? Probably the most isolated.
Yeah, so there's they're actually claiming there's structure within the Orkneys.
It's a mixture of groups.
Yeah, so they have 2 clusters in the Orkneys, and I think that they are thought to differ in the amount. I think I think the Orkneys have got a strong contribution from Scandinavia, and I think that the 2 clusters differ in how much of that they have. I can't remember the details on that. But yeah, so if you're a wildlife manager, the question is, like, which of these are ESUs?
The Orkneys.
Yeah.
Do you have an idea what's going on in the north? Because obviously very different from the The blue one.
What's that?
In England.
The north of England.
The blue cluster. Very different from the rest of the country.
Yeah.
The Midlands.
I don't know enough English history. I mean, I guess. I think they probably talked about this in the paper. I don't remember. So I guess this is like Liverpool area, the Beatles and all that. Yeah.
What if you don't have a full genome? Can you do it with maps, every map?
So you can do it with contigs. Yeah, I think. Yeah, there's no reason not to do it with contigs. Right? Okay. Okay, so we've got about 20 minutes left, and I was gonna have some question and answer time on structure, but I also had, this took me longer than I expected. I also have some stuff on learning about trees with gene flow between populations. So shall I go to that? All right.
Can I ask a question?
Yeah, go for it.
So in expanding to genomic data, what are your thoughts on kind of trying to get rid of both sides structures?
Oh, I guess I don't worry about it. So structures. So I mean, it should be generally robust to that, because if you have. So usually when there's selection going on, it will, it'll, like, if anything's going to kind of enhance the signal, so I mean, if you've got selection that's differential between different populations, then it'll increase frequency differences, and that would just help. Secondly, the fraction of loci in the genome that show really strong signals is pretty small, so like neutral stuff will tend to drive the signal. So, yeah, I don't yeah.
So I had a project where for K of 3 it was basically getting two answers. One was really high probability in the biological sense, the other was really low probability in the negative sense. And I solved it by using multiple pop-alphas. But what I was wondering is, if you get something like that, is it valid to say, I'm going to, do 40 runs and use 20 with the highest probability?
Yeah, I think that's reasonable. So it sounds like this. So the convergence is stochastic, and so sometimes it gets stuck in like in the wrong peaks, and it sounds like maybe that's what was going on here. But you can recognize that to some extent by differences in the probability of the model, and then I just discard those. This actually gets to the point of really long runs. So, I think it's much more effective to use your compute time to do a whole bunch of runs that are shorter than one really long run. And the reason is that, so structure. I mean, the Markov chain Monte Carlo can explore local peaks, but it's extraordinarily unlikely for it to be able to drop out of one peak and get to another peak. So even though, like, in theory it can do that, you might need to run the program to the end of the universe to be able to drop out of one peak and get to another peak. So so a long run, if you run structure for 50 million iterations, you're just exploring around the same peak a lot of times. Whereas if you start it 10 times from random starting points, then it gives you a chance to get into different peaks and see whether there are things that you've missed. So, yeah.
I actually have a question about that. We sometimes see that people say to use out of 10 runs, we'll use the 8 with variation. And they just drop some runs there.
So the criterion that we've used before is similar to what you were saying, that like some runs with lower probability. Like if it looks like there's multimodal stuff going on, and we've dropped runs with lower probability. The variants are, well, part of the problem is that there's no real theory to guide here. So all of the theory from CMC is asymptotic. It's, you know, like basically like if you run these Markov chains to infinity, then they do what you want. And you know, but we can't run them that long, so you know, so then it's a little bit ad hoc how you deal with these sorts of things. But maybe I need to know more about details. All right. So let me just talk briefly about population histories. So this is work with Joe Pickrell, who was a student of mine. He now runs his own lab in New York. And so we were interested in trying to say more about historical relations among populations. And of course, you know, phylogenetic trees are really central to how we think about historical relations. There's a phylogeny of the primates. Here's a picture from Darwin's notebook. And this idea is so ingrained in our thinking that if you search on Google image for that, you find out that it's a popular tattoo. Not that I necessarily recommend that. And so when you're looking between species, I think phylogenetics make a lot of sense. But within species, it's not clear that the phylogenetic tree representation is a sensible thing to do at all. So here's a beautiful figure from a paper about dog breeds. They've got this, you know, beautiful looking tree. But do we really believe that this tree reflects reality, given that all of these breeds can interbreed? So if there's been ancient migration, admixture, mixture, then a bifurcating tree like this might not be a good description of population histories. And it's clear that there's tons of gene flow in many cases between populations, between continents, and so on. So here's a blow up of part of the tree. So I showed you before this data set of 52 human populations from around the world. And so this is a tree that was made in a 2008 paper using about 600,000 SNP markers across the genome. And so I'm just blowing up part of the tree so you can read the labels. And so the tree looks beautiful, right? So you've got the Sub-Saharan African group here, you've got the Middle Eastern clade here, you've got the Europeans. And then, if you look at the structure plot from the same paper, the Middle Easterners look completely admixed. So the Middle Easterners here, they've got a Middle Eastern component, but the same individuals have got this European component, and blue here is as a Central Asian component. You can't see because I cut off the figure. So it's clear that the structure plot is suggesting that there's tons of admixture. The tree is giving us this beautifully simple picture that doesn't show any admixture. So what are good ways that we could develop for building trees that reflect these extra complications? So in thinking about this, we thought that often a graph might be a better representation of the history than a bifurcating tree. And so a simple version of a graph would be something like this, where so the black lines here are showing kind of the main lines of descent, but this population 3 has got some of its ancestry coming down this branch as well. So we'll say it's got a fraction W of its ancestry comes down this way, and then 1 minus W of its ancestry comes down this branch. So this population, when you do the tree, without these migration arrows, it'll be kind of arbitrary where it gets clustered in, whether it gets clustered with population 2 or with population 4. We'd like to be able to estimate this directly from the data. So the practical question then is how might we estimate these historical graphs? So I'm going to assume that we've got SNP data for tens of thousands, hundreds of thousands of SNPs in each of a number of populations. For this purpose, I'm going to assume that we know what the populations are. So this is different from structure. We're going to use the labels. You know, you could run something like structure around mixture first to see if the labels make sense. Okay, so there's some classic theory on methods for estimating trees from allele frequency data. Some of this goes back to Joe Felsenstein and Luke Cavalli-Sforza back in the 70s. And so their model was that you can think about the allele frequencies. You can approximate what's happening to the allele frequencies along the branches of the tree by a drift model, in which drift is modeled by what's called Brownian motion. And so what Brownian motion does is it says that the allele frequency distribution, that the change in allele frequencies along each branch, you can approximate that as a normal random variable. So it's got a normal distribution. And if you think about genetic drift, you might convince yourself that that's probably a sensible model over short time periods, although it's clearly not. It's clearly not an exact model because allele frequencies are bound between 0 and 1, but the normal distribution takes on all real values. But anyway, if you make that approximation, then the math all works out very nicely. So this Brownian motion model leads to a simple covariance matrix. So let me just say what the covariance matrix is for a moment. So imagine we've got a list of estimated allele frequencies in each population. So now we can take any two populations and compute the covariance in allele frequencies in those populations relative to the average. So it's like a measure of how much similarity is there in allele frequencies between two samples relative to the set as a whole. And under this Brownian motion model, we've got these parameters which are kind of which are reflecting the amount of drift that's going on down each branch. And so so this is the covariance matrix. So so population 1, x1 against x1. So that's the variance term for how much drift there is in population 1. So it turns out that there's another normalizing factor I'm not showing here. Depends on allele frequencies. But that variance is going to depend just on the amount of branch length there is between the root and population x1. So that'll be the sum of these two branch lengths, C2 plus C6. And then if we look at the covariance between these two populations, the covariance is equal to just the shared branch length, which is C2. And then the key bit is that for populations that are on opposite sides of the root, there's no covariance at all because they don't have any shared branch length from the root. So you get, for example, population x3 against x1, this entry here, this covariance entry is 0. And so you get this very nice block structure in the covariance matrix. And one of the nice things about the second nice thing about working with this model is that it takes a huge amount of data and boils it down to this covariance matrix, which is relatively simple to work with, and it makes the computation practical. So basically what we want to do is, so in the old work in the 1970s, they didn't phrase it exactly like this, but their goal was to try to find trees that are consistent with this kind of covariance matrix. So now, what we want to do is to extend this to add in migration arrows. And then what this migration arrow does is it adds in all of these extra terms here. So for example, everything in red here is a factor that's being added in as a result of this migration arrow. So for example, you get covariance now between population 3 and population 2. So x3 is correlated now with 1 and 2. And remember, those were zeros before. And so basically, if you take a graph like this that's got this extra migration arrow, then there's no single tree that's consistent with this. and so you have to add in migration arrows to make the graph be consistent with the observed covariance matrix. So the next step then is to take the covariance matrix and tackle this inverse problem of figuring out whether migration graphs are consistent with that. And that's kind of a challenging problem, and we have something that works for that now. So let me just show you some examples. So, so I showed you before that we think there's a lot of migration going on in the Middle East, and so now this is a representation of the migration graph for humans. I'll show you a blob so you can read the names, but basically the black lines are kind of the backbone of the tree, and then all of the orange and yellow arrows represent migration arrows, and they're defined to have weight less than 0.5, so that they're always weaker than the black lines, which are the backbone of the tree. So you can see there's tons of migration going on here from Sub-Saharan Africa into the Middle East. And here's the blow-up of that. So here are the Sub-Saharan populations, here are the Middle Eastern populations. And you can see that we're estimating lots of gene flow events from Sub-Saharan Africa into here, and also the Middle Eastern European populations. I've also done this for the dog data that I showed you the tree for at the beginning, and again you can see that there are tons of migration arrows on here. We stopped drawing them at some point because there are, if you put too many on, it's hard to see anything. Let me show you a couple of one interesting example. So there's a clade here on the tree which has got wolfhound, greyhound, and whippet on it. So these are all sighthounds, but other sighthounds are scattered on the tree. So Italian greyhound is up here, and borzoi is here. So at first we were a little bit mystified by this, but here's sort of a blow up of that. So this is the main sighthound clade here. So these are wolfhound, greyhound, whippet, and then Italian greyhound is next to miniature pincher, and borzoi is next to Eskimo dog and Samoyed. So this is what borzoi looks like. So it's got the same kind of shape as the other sighthounds, but it's added this massive white fluff, and that's where it gets it from. This is Italian greyhound. So again, it's the same kind of shape, but it's tiny. It's about 6 kilograms instead of these 10 to 50 kilograms, and so we think it proposing gets small size genes from miniature pincher. So that's a model that we've developed for learning about these more complicated admixture graphs. We can also ask what kind of signal selection would produce in these models. And so this is maybe getting back to your question a bit. So we have this idea that selection should produce unusual allele frequency distributions of particular SNPs. So you might think of this as seeing that there are distantly related populations that share SNPs in a way that's really surprising given the overall genomic background. And so one thing that we can do then is for each SNP just to simply ask how well its frequency distribution matches the overall covariance structure. And so on this, the y-axis is kind of a measure of deviation away from the background global distribution. And the strongest overall, the strongest outliers in the whole genome are on chromosome 15. You see them here. And so when we look at which breeds have those, so these ones have got one of the alleles at 100% frequency. These ones have got it at 0% frequency. Any guesses? Yes, that's right. So they're adjacent to IGF-1, which has been shown to play an important role in body size in dogs. And in fact, all of the top outlying regions in dogs are associated with morphological phenotypes that breeders have targeted. So these include body size, coat color, coat length and texture, snout length. So interestingly, in humans, the picture's a lot less clear, and only 2 of the top hits are currently associated with known selective phenotypes. So one is skin pigmentation, and EDAR seems to be associated with hair morphology and sweat glands, although I think it's a little bit less understood exactly what specifically it's doing. So we think that overall there's a lot less signal for these very, very strong selective events at individual loci in humans. And in humans, even the most discordant SNPs are, to a large extent, they reflect neutral population structure. So this is the distribution of one of the SNPs that's involved in skin pigmentation. It's associated with lighter skin pigmentation, the red allele is. And it actually, its global distribution matches very closely to the distribution of one of the major clusters. And so based on this and a bunch of other things, we think that most adaptation in humans is much more likely to be polygenic. So it's acting on so if selection's acting on complex traits, then what it will do is it will tend to shift the allele frequencies at hundreds or thousands of markers at the same time, but only by small amounts, so that it'll be very difficult to pick up individual loci by sort of traditional selection scans. So this is sort of a distinction between the hard sweep model, where you have a single marker that sweeps through and clears out variation in one region, compared to polygenic adaptation, where you've got small shifts in allele frequency spread across many, many loci. And I'll just end with one example quickly. So we now know that height is incredibly polygenic. A recent study identified about 700 loci that are genome-wide significant for height, using 250,000 people. And it's known that on average Northern Europeans are taller than Southern Europeans and other human populations. And so there's quite a nice study that looked at selection on height loci. And so the hypothesis here is that the European differences in height may be driven by adaptation in Northern Europeans, although the nature of the selection pressure is not exactly clear. So what they did was they took the 700 or so loci that have been identified as affecting height, and then for each of those they took the difference between the tall allele frequency in Northern Europe minus the tall allele frequency in Southern Europe, and so the model is that if the height difference between Northern and Southern Europeans was purely environmental, then you'd expect that there's not really a net change in allele frequencies. But what they actually saw was that the tall alleles are consistently going up in frequency in Northern Europeans relative to Southern Europeans. And so it's only about a 1% effect. But for the most strongly associated, so the x-axis here is ranking SNPs according to their effect sizes, and then the y-axis is the difference in allele frequencies between Northern and Southern Europe. And so for the most strongly associated loci, the height is on average about a 1% difference in frequency between Northern Europe and Southern Europe, with the tall alleles going up in frequency. And then as you go to less and less associated loci, this difference is dropping back towards zero. So I think this is pretty compelling evidence for height that there's polygenic adaptation. It's a tiny selective effect spread across thousands of loci. One thing that we're working on a lot in the lab now is trying to extend these kinds of methods to a lot of other traits. And there are some technical challenges with other traits that we're dealing with. So anyway, I'll end there. I'm out of time. Thank you.